Hello, my name is Mr. Chipman, and I teach biology, AP Biology at Murray High School in Murray, Kentucky. And this is 3.4 Photosynthesis, Part 1. Decided to divide photosynthesis into two parts for my own classroom, just for a cognitive load sort of thing. It's a lot, right? It's a lot of weird stuff, and it's pretty heavy. And so I think it's a good thing to divide in half. Part 2 will be around shortly. Basically what I've done is I've divided it into the two different reactions that we're going to look at. And this first video is going to be more of a overview of the whole process as well, so it'll be a little bit longer. So here we have the overview of photosynthesis. First of all, what is photosynthesis? It's using energy from light to make glucose. Or if you wanted to be a little more specific, you could say using energy from light to fix CO2, atmospheric CO2, in order to make glucose. Right? This idea of carbon fixation is taking unusable carbon from the atmosphere in the form of CO2 and making something usable out of it, which is glucose. Some other things are going to be necessary for that. Water is a necessary reactant in this process, and oxygen is going to be what we're going to call a byproduct of this reaction. Obviously, oxygen is important. We love the fact that plants give off oxygen, but for the plant's case, it's not going to be a necessary product coming from this, even though plants can use the oxygen that they make in order to do cellular respiration. And you can see some other things that are going on there. They're cycling in the middle of this. This is a picture of a chloroplast. You have some NADPH, and some ATP, and some ADP, and some NADP plus. So those things are also important. Let's look at just some general definitions of these two reactions. And I think it's good to sort of have these definitions of these reactions as we get into the weeds on them. It'll help you to sort of anchor your thoughts, so to speak. So on the left, we have the light-dependent reaction. In the light-dependent reaction, of course, you have light, and light is used to break apart water molecules, H2O, on the thylakoid membrane. So this is happening on the thylakoid membrane. It is used to break apart water molecules, and those water molecules are going to provide a steady stream of electrons, right? And those electrons are going to be picked up by electron carriers NADPH and carried to the Calvin cycle. ATP is also made in the light-dependent reaction, and so ATP and NADPH are the primary products of the light-dependent reaction. This is the thing that is being made that is necessary for the next step of the phase. Oxygen is going to be a byproduct of this reaction. On the right, you have the light-independent reaction. AP literature will not use that terminology at all. I haven't seen it in AP literature. I think it's better than the term that the AP uses, which is Calvin cycle. It's fine. Calvin cycle is used ubiquitously in the scientific community. I just don't think it's very descriptive, right? A light-independent reaction is more descriptive. However, we're going to use the Calvin cycle. Calvin cycle happens in the stroma. The stroma is the fluid portion of the chloroplast, and what's happening in the light-independent or the Calvin cycle is energy from NADPH and ATP is being used to fix carbon, and glucose is going to be the product of this reaction. Notice that ATP and NADPH are used up in this reaction, and the ADP, which represents the used up form of ATP, and NADP plus, which represents that NADPH having dropped off those electrons that were picked up in the light-dependent reaction, are going to go back to the light-dependent reaction and pick up more things. And so you can see that as a form of energy coupling and energy or reaction sort of cycling as well. Moving on, chloroplast. Let's talk a little bit about chloroplast anatomy. Some of the terms that you're going to see in the exam, the two that we talked about, and just briefly, the thylakoid membrane and the stroma. The thylakoids are these little discs that you see individually stacked inside of a chloroplast. They look like little Mentos or something, right? And on this membrane is the light-dependent reaction. Tons of surface area to volume sort of ideas here, right? Lots and lots of thylakoids in here. A stack of these thylakoids is called a granum. I've just seen it on the exam, seen it on the FRQs. And so it's just a word to have in your back pocket. Just so when you see it, it doesn't bother you, but it's, not that important. Stroma is where the Calvin cycle takes place, and it is just the fluid out here. Notice the chloroplast has a double membrane as well. We've talked about that in other units. And then let's talk about chlorophyll. Chlorophyll is the green pigment found in plants, and chlorophyll is going to be very important in this. Pigments are light-absorbing molecules, and so just a real brief understanding of this. When we say that chlorophyll is a pigment and that pigments absorb light and that chlorophyll is green, sometimes we immediately think that what's being absorbed then by chlorophyll is green light. That is not the case. Chlorophyll absorbs all the other lights. It reflects green. The reason that you see green when you look at chlorophyll is because it's reflecting it back to you, right? It is absorbing all the other light colors on the spectrum and absorbing those as energy, reflecting green back out into the atmosphere, and you see that green, right? That's an important distinction. Don't have a graph here to show you. There's a very common graph that shows the light spectrum on the x-axis and absorbance on the y-axis, and you can see absorption is really high in the red spectrum, the blue spectrum, but really low in green because it's not being absorbed, it's being reflected. Why is chlorophyll important? Well, let's zoom in. All kinds of different sorts of chlorophylls here. AP is not outside their realm to talk about chlorophyll B or chlorophyll C3 or anything, but it's all the same thing, essentially the same thing. In the center of every chlorophyll molecule is a magnesium ion. And this magnesium ion, it's a magnesium atom. And magnesium atoms ionize, and when they do, they lose 2 electrons, right? And it causes magnesium to have this sort of ionization. Those 2 electrons are extremely important, right? And we just a brief understanding of electrons is when they get energized, they spin faster, right? And they spin further away from the nucleus. Well, what happens here is when sunlight hits chlorophyll, it causes those 2 electrons to sort of release from the magnesium, right? And then magnesium doesn't have any more electrons. Well, it needs more electrons. Where is it going to get more electrons? It's going to get them from water. We mentioned that earlier. Water supplies electrons. And these electrons, these 2 electrons, are really going to be the story of photosynthesis and cellular respiration. We're going to see these 2 electrons travel very far and wide and end up back with water at the end of the day. It's kind of like a little love story or something. All right, so you have magnesium, center of every chlorophyll molecule, and those magnesiums are very important, maybe the most important atom in the world as far as we're concerned, as far as living things are concerned. Without chlorophyll, we don't have glucose. Without glucose, we don't really have any energy. So it's pretty important. And we need to understand this idea of electron carriers. Electron carrier is a molecule that, you're going to be surprised to hear this, carries electrons. And it does this by, you're going to see this oxidation reduction sort of thing. We don't really need to be in the ins and outs of the oxidation reduction, but just understand that when you see that something is reduced, it is talking about its charge. And how would something's charge be reduced, become more negative? How would it become more negative? By gaining electrons. And so when NADP plus is reduced, it is going to become NADPH. It's picking up those 2 electrons that magnesium let go of, that water when it was broken up, got rid of, right? And then it's going to go and reduce something else. It becomes oxidized and it reduces something else. And that exchange of electrons, we're going to see that occurring. And that exchange of electrons is an exchange of energy. And that energy exchange is going to be used to power systems, which are going to be used to make ATP. Very important to this whole discussion. Speaking of this discussion, here we go. So the 2 reactions for photosynthesis, as far as AP is concerned, there's a lot of emphasis on the detail of light-dependent reaction. And when I say the detail, it's sort of the broad brush detail that we're going to get into. whereas there's a whole lot less emphasis on the Calvin cycle. And so you're going to see that the video for the Calvin cycle is going to be significantly shorter than this one. So here we have a picture of our chloroplast. Here we have this little zoom in window. Love it. And you have this is a zoomed in picture of the thylakoid membrane. And a couple things just to note here. The PS2 and PS1 also being defunct gaming systems, but now also, in this particular context, are photosystems. Photosystem is a group of pigments. Pigments are light-absorbing molecules. The primary pigment involved in here is chlorophyll. So when you think of photosystem 2 and photosystem 1, just think of this is where the chlorophyll is, right? This is where light is being absorbed. Photosystem 2 comes first, unfortunately, in the sequence, just because we like to make things as difficult as possible. And then you see light being represented by these little lightning bolts. Love this picture. So I'm going to go through the sequence of this, and I'm going to start with light being absorbed in photosystem 2. When light is absorbed in photosystem 2, that magnesium that's and in reality, what I'm going to want to do here is I'm going to want to put a little 2 here. See if I can see my dot. It's really small. There we go. I'm going to want to put a little 2 there just to help us to remember this is 2 electrons. All right, and so light, I lost my dot. I have to have my dot. Okay, so light is absorbed on photosystem 2, and then that light absorbing causes those 2 electrons for magnesium to release, and it is released into this area called the electron transport chain, or the ETC. ETC is a series of proteins, right? And one of them happens to be a hydrogen pump, or a proton pump, excuse me. And so we're going to get to that in a minute. It's going to go through this electron transport chain. What do you think the electron transport chain does? It transports electrons, right? This is going to be where those oxidation reduction reactions are occurring that we were talking about earlier. So magnesium is let go of its 2 electrons. It has no more electrons. Where's it going to get more electrons? That's where water comes in. So here we have water coming in. Water is going to interact here with photosystem 2, and the energy from the light's very good, powerful, very clean energy, is going to shatter this water molecule. Water molecule is made of several things. It's made of an oxygen, and it's made of 2 hydrogens, and those 2 hydrogens are each made of a proton and an electron. So we have 2 electrons here, right, that are going to be shattered, and it's going to continue this electrons going into the electron transport chain. Those 2 protons are going to be important as well. We're going to talk about those protons. They're going to start to build up a concentration gradient inside the thylakoid, and that oxygen here is going to be released as a byproduct. And so this is where oxygen is released as a byproduct. And so those 2 electrons are going to go through the electron transport chain here, and they're going to power the pumping of protons into the thylakoid membrane, right? And so this is going to build up a concentration gradient. Remember, this would be active transport, because we're pumping protons from a low concentration outside the thylakoid to a high concentration inside the thylakoid. We've talked about this briefly in Unit 2, and we said that if we can build a concentration gradient, we can use that gradient to do some sort of cellular work. And so we have a concentration gradient being built up inside the thylakoid, whereas there's a positive or a high pressure of protons inside the membrane and a low on the outside. All right, so that's being built up. Those electrons, tired from their pumping proton work, are going to be re-energized here at photosystem 1. And then right outside of photosystem 1 on another little protein that doesn't have a name as far as we're concerned, those electrons are going to be picked up by NADP+, and NADPH is going to be formed, and now NADPH is going to the Calvin cycle. So these electrons that originated over here in magnesium and water are now being taken full of energy to the Calvin cycle. All right, so what are we going to do with our concentration gradient? In our concentration gradient, we're going to go from an area of high concentration inside the thylakoid to low concentration outside the thylakoid. Those protons are going to they're going to pass through this giant protein called ATP synthase. Look at the word ATP synthase. Looks like synthesis, except for it's an enzyme. It's an enzyme that's making ATP. How's it doing that? It's using this concentration gradient that's being built up. And the energy of that proton passing through ATP synthase is going to create ATP from ADP and a phosphate. This little molecule is basically going to spin around, right? It's almost like I like to think of it as like a salad spinner, right? You apply energy to the salad spinner, it spins around, and that energy throws water off the lettuce leaves, right? Well, the energy here is not going to throw water off lettuce leaves. Instead, it's going to put a phosphate onto ADP and make an ATP. Now we have NADPH and we have ATP that have been made in this process. And if we go all the way back up here, we see that is very important, right? That is the business end of light-dependent reaction. Oxygen being let off is just a byproduct, right? It was let off when water was split, remember. But these two things go into the Calvin cycle, extremely important part of this, because they represent the sun's energy now converted into chemicals. I know that's a lot. If you have questions, ask in the comments, please. Let's look at a practice problem. A student claims that the thylakoid membrane plays a key role in ATP production during photosynthesis. He's right. Which of the following best supports the claim? Okay, so what we're going to do down in ABCD is we're going to look for something that tells us that about a concentration, the thylakoid membrane being used to build a concentration gradient, right? Because that is how ATP is being produced. Let's go find that. A, thylakoid membrane contains DNA that codes for ATP synthase. Why does that even matter? It's not true. Thylakoid membrane is a site where oxygen is absorbed from the atmosphere. It's not what's going on. Thylakoid membrane maintains a proton gradient that allows ATP synthase to phosphorylate ADP. We like that. Phosphorylate is just adding a phosphate to it. We're going to look at D just in case, right? Sometimes we're going to do that. I know some of you guys need that. Thylakoid membrane reduces glucose. It's not what happens here. C is the correct answer. Again, this is hard. If you have additional questions, please ask down in the comments. I hope this was helpful.